I will present here a little bit the tools that we are doing. We are not already in the Deuterium field, but we hope through collaboration we will manage to apply our methodologies to demonstrate these effects of Deuterium. So I will present first what we are doing and some applications, and after how we can apply to this field. So our team is working in metabolism. Even when metabolism was not at all in fashion, we resist and say metabolism will come again. And now we are glad to see that metabolomics and fluxomics are key elements in the systems biology approach. So in studying the integrating all these omics data in the field of models to try to understand metabolic reprogramming, pathological metabolic reprogramming associated with a lot of multifactorial disease. In these fields, we really believe that metabolic and fluxomics are key for understanding what is happening at systemic level, and key players in all these efforts ongoing in several labs nowadays in constructing in silico simulators to analyze and understand the metabolism of the cells, and especially its dynamics. So we really believe that metabolics and fluxomics are so important because we are convinced that are the more close to the phenotype and have a high capacity to really predict the phenotype, because metabolics nowadays is already enough evidence to believe and has been demonstrated that not just the end point of a hierarchical instructions from what is in the genes, but are also the driving force affecting which genes are expressed and which genes are not expressed. So we really believe that these centuries will be the rush of metabolic studies, and that multifactorial disease that is still killing so much people really we will advance in found therapeutical drugs and in cure, studying more in deep metabolism and understanding metabolic reprogramming and especially dynamics of the metabolism. It's for this that we are so interested in metabolic fluxes, because it's like to have a photo or to have a video. So you have no metabolic fluxes, you have the video, and we will really know better what is happening with the video than just with a static picture. So we have been in this field for a lot of years. We started pioneer work with our friend Laszlo Boros from a lot of years ago, and we are convinced that carbon-13 use of labile substrate as a tracer is really a wonderful tool to understand this metabolic reprogramming. These techniques are not so complicated. You need to incubate, for instance, cells or animals or what you want to tracer with carbon-13 labile substrate and follow the incorporation of this level and how the level is incorporated in different intermediate metabolisms combined with flux balance analysis permit you estimation of metabolic flux map. And you can interpret this data either by using analytical format, what is in the field is named MIDA analysis, or integrating through mathematical software, and we develop software for this purpose at the end to have a picture of metabolic flux distributions. So the point is that when you have all these data, if you can do simulations, and if you can ever integrate all this data with kinetic models, take it into account regulation of the enzymes, and this gives even more power to really have in silico metabolic simulator. And you can use to analyze and understand metabolic adaptations, supporting cell function or supporting pathological reprogramming in cancer, for instance, and design metabolic interventions to complement targeting metabolic adaptation with established ongoing therapies. And of course could be other application at biotechnological level, but we are interested in health application to found complementary target for multifactorial disease. So this is a classical experiment. You incubate the cells with, for instance, typically glucose 1,2 labeled, but also glutamine labeled and so on, lactate labeled. You do the culture, you compute biochemical levels, how glucose, lactate, amino acids are consumed. You can do this with a lot of techniques. And after you can compute isotopomeric distributions, and in our lab we do using GC-MS, and after we feed all this data to have quantitative flux maps. And this, at the end, these techniques are based in that, depending the pathway, for instance, that glucose follows to arrive to lactate, for instance, the level that you will observe in lactate will be different if has followed directly glycolysis breakdown in triosas, in which the two levels became stacked together, or if they go through oxidative pentose phosphate pathway. In this case, the first level is lost, and you have lactate with only one level. So this type of techniques also can be applied to other metabolites at the level of glutamate, permit to trust that if they came for pyruvate carboxylase or pyruvate dehydrogenases, you can compute ratios looking at the levels of glutamate. So are really powerful tools to map the metabolic network activities and which pathways are enhanced or decrease it in cancer. So now we are working in this model. We have developed disease sorting software, and we are coupling to a really detailed kinetic model of these reactions, and we plan in the future to, and we are working on this direction, to couple also to a model of mitochondria ROS production that we have for the moment separate these models, but we are trying to coupling all together. The point is that when we apply in cancer, that has been our main work, has been applying these techniques in cancer. The idea is that cancer cells has a robust metabolic profile, but everything that is very robust has fragility, so unexpected perturbations and this robust metabolic profile can show these vulnerabilities that can be exploited, and could be therapeutical windows in which some pathways, like pentose phosphate pathways that are enhanced in cancer cells, can be targeted, and the cancer cell will be more vulnerable than the normal cell to these unexpected perturbations. This is the idea, and this is what we have been working. And these techniques have been used in our lab and in other labs for a lot of applications. For instance, to understand better metabolic reprogramming after gene knockdown, overexpression, after drug interventions, to compare metabolic phenotypes of different cell types. Now we are working a lot to compare cancer stem cells versus cancer non-stem cells. Metabolic reprogramming could be useful for stratification, select biomarkers, select a target at metabolic level that you can select a population of patients. Because when you translated all these changes in flux maps, you can really do targeted interventions according pathologic metabolic reprogramming of different subpopulations. And even we have been with these techniques, we have been provide evidence that metabolism is quite more compartmentalized, that not only compartments are the bones, mitochondria, nucleus, but there are also supramolecular organization in cytosol that generate different pools of metabolites. And if not, sometimes you cannot really explain the levels without assuming this compartmentalization. Some things are much more complex than we thought in the beginning, but this is good because there's room for understanding and for developing specific strategies in targeting specific pathological metabolic reprogramming. The idea is we need a rational design of new therapeutic combinations with targeting also metabolic pathological reprogramming, and for this we really need a deeper knowledge of metabolic network and metabolic fluxes. Now, after I will try to show some examples of application of this technique. This is early work on HC29 that we started years ago with Dr. Lee and Dr. Bolos. We focus in these studies altogether that a pentose phosphate pathway is really enhanced in cancer cells. And here you see in this very interesting model that is adenocarcinoma model, because these cells can grow undifferentiated, but you can differentiate them with either butyrate or tricostatin, and we study this in deep, these metabolic changes, and we see that when the cells differentiated and adhere a metabolite, a phenotype close to the mucosal cells of colon, they really decrease ribosome production, and they really decrease lactate production. So this ribosome enhancement in undifferentiated cell versus differentiated cells make us to go in deep in the interest of targeting pentosas in colon cancer. We go to develop some new inhibitors for human transcatolase. We have go also to study how is the transcatolase at levels in an isoenzyme profile, because transcatolase has two isoenzymes. And we have analyzed it in real slides of tumor samples, well classified according to their stage. And what we have seen is that stage 3 tumors present the highest level of isoenzyme transcatolase like 1, that is preferably this isoenzyme is present in cancer cells. And this increase of transcatolase correlate with regional lymph node affectation of patients. And we also curious observed that transcatolase like 1 decrease when already there is distant metastasis appears. So probably it's important for starting metastasis when metastasis has been established, the primary tumor not need so much this transcatolase like 1. So the idea here is that from a study of the metabolics, we shift to conclusions that these enzymes would be important, and after when you go to patient, you see that really correlation with one of the isoenzymes that is overexpressed in tumors with a stage of the tumors. And this is one of the things that we have done, and I present here to see the type of studies. Another study that we have been done is the effect of nutritional interventions. This, I think, would be interested Agostini, because we have been working with polyphenols and grape antioxidant fiber. This is extracted for the part of the grapes that not go to the wine. So this is a vast product. From there we extracted a fiber that has antioxidants attached, and this is very natural thing. And we show that it's very useful for cancer prevention and at the level of animal models. And the idea is to show this study and we also will show that now we are something that are going that also apply to these strategies for another disease that is chronic obstructive pulmonary disease. So these are part of polyphenols and nutritional polyphenols and cancer. We started from in vitro studies with cells that we supplement with different polyphenols. In particular, I'm showing here epicatechin-galate supplements to the cells, and these are again this colon cancer model. And what we observe is that epicatechin-galate, one of the polyphenols present in grape and in tea, what do is that really decrease glucose consumption, lactate production also is decreased, the novel lipid synthesis from glucose is also decreased, and also ribosome metabolism is decreased. So we were excited with these cell results, and we have go to animal models, but we decided that to the animals we will give something that is nutritionally natural and is available, and we extract this from from what is not used for the wine production, because you know Catalonia and Spain are good wine production. This part that you have tones that is not used is what remains after extract for the part for that go to the wine. Is really rich in fiber and in natural antioxidants. So from these extractions, from this vast material, we prepare this grape antioxidant fiber, and we give first to normal mice, and we do a classical experiment of providing to normal mice, not tumor these mice, to see if we can show that could be some prevention, some indications that this could be preventive of colon cancer, and with the idea after to give to a colon cancer animal model. And so, this study we have been in collaboration with Dr. Ulrich Gunter, that performed the NMR experiments in this study. So what we have done is a classical study of giving for gastric garbage this fever for several weeks, and we follow the mice, and we follow the weight of the mice, and after, at the end of experiments, we extracted the colon, and we analyzed what is the microRISE, what is the change in the transcriptomic profile after these weeks of providing this supplement in the diet. And what we see is a regulations of the genes related to antioxidant activity and xenobiotic metabolism. And we see an overexpression of endogenous defense systems. So it's like we think from after looking at this that perhaps they produce a mild pro-oxidant effect that may be a real benefit, because we know that ROS at low concentrations are signaling, at high concentrations are damaging, and perhaps in some manner they stimulate the natural, according to the results of this is microRISE from the intestines, this stimulate the natural defense at the local level of intestinal tract. So we also analyze other interesting. We also identify other interesting things that upregulation of gene related with apoptosis, tumor suppression cells, cell differentiation, energy metabolism, immune system response, amino acid metabolism and regulation, cell cycle and its regulation, and the regulation of gene related of tumor development, proto-oncogenes, lipid biosynthesis and regulation, glucose metabolism, TCA cycle. But here in TCA cycle, we need to be careful, because what we see is a decrease in PTK4, but PTK4 is a kinase that phosphorylates pyruvate dehydrogenase, and pyruvate dehydrogenase phosphorylated is less active. In fact, increasing this gene, what you are doing is increasing pyruvate dehydrogenase activity. So paradoxically, what you are seeing is increased TCA cycle, because you are repressing the natural inhibitor of pyruvate dehydrogenase. Okay, and this is the part that in the laboratory of our collaborator, Dr. Ulrich measured, and I think Agostino will be interested, because this diet reduces glucose, lactate, tryptophan, and increase ketone bodies, okay, and some amino acids. So I think this is perhaps interesting. I never thought about this before being there, but could be interesting. A coadjuvant to ketogenesis diet, because I think we are going in the good direction with this natural food supplement. So if we have been trying to relate what Ulrich see in these samples with our microRISE analysis, and so this some genetic decrease, some transcript decrease of the gene that we see could correlate with high blood amino acid levels. PTK4 decrease results in PTH activity increase, more acetyl-CoA could be related with these blood ketone bodies, because what is interesting in this study is that NMR that Ulrich has done is in blood, and the change that I am reporting in transcriptomics are in the colon where so what we see the observations are concordant. So the fact that this antioxidant is at the end is fiber with antioxidants attached. So we know that fiber decrease carbohydrate digestion. So it's correlated with the blood glucose levels that Ulrich see, and also to the lowering blood lactate level. So we were excited with the results. So we buy this other model that is the same mice, but these are a model commercially available that are APC minus mice. That these mice is a model that is used for colon cancer, because it's a model that spontaneously make a lot of polyps in intestines and colon in a few weeks. So we use this not spontaneously producing polyps mice and to do the same treatment with antioxidant fever. And what we see is that when we measure the intestinal polyps, because mainly this model produces intestinal polyps in the small intestine, we see decrease in the total number of polyps, but also the decrease was in all the different regions of the intestine, and not only the number of polyps decreased, but also the size of polyps decrease, and we see a decrease in all the regions and in all the different types of polyps according to the size. Okay. And the decrease was really very, very important, was 76%, and this perhaps optimizing the doses of fever even can be improved. No? So this is what we observed, and I am happy to see that this correlates with the keto, that this diet also is increasing ketone bodies, because this has a very interesting correlation that I not thought before coming here. No? So this is what we have been doing, and some applications. And just I want to comment also that now we are connecting all these metabolic modeling efforts that incorporate kinetic details of the central metabolized enzymes to have a template to model, to do quantitative flux map from carbon 13 distribution in the intermediates, and also studying the influx of influx and outflux of the main source of carbon, like glucose consumption, lactate production, glutamine consumption, glutamate production. And we have been doing joint efforts there also in a framework of MetaFlux that has been one of these tiny networks coordinated by Dr. Ulrich Gunther, sponsored by European Commission. And now we are connecting this to our model. We have a very detailed model of ROS production in mitochondria. And we have demonstrated some years ago that for their natural mechanism of functioning, the complex 3, complex 1 can operate in a B-state model in the manner that they can produce high ROS or low ROS, and you can arrive to this state of high ROS, and after in hypoxia, but after normalizing oxygen conditions, not always you not return to the low ROS production levels, which is well known for years and is a problem in transplantation of organs, for instance. And what we demonstrated that inherent to this could be the fact that stability exists in ROS production at the level of especially of complex 3, that is what we more study. So this is what we're working now, trying to couple our metabolic models with ROS production models in mitochondria. And in addition, we are trying to integrate this layer of information to what is now also in fashion, that is genome scale metabolic models that are mainly construction for transcriptomic data on metabolic related enzymes, and being huge RECON2-1, RECON2, very huge genome scale metabolic maps. So we really believe that introducing to these metabolic maps, mainly constructed in genome and transcriptomic data to include metabolic fluxes and as a layer, and all a computer with this carbon 13 experiment will really have a more powerful tool to really understand metabolic network reprogramming in multifactorial disease like cancer. And we think that all these strategies and tools that we are using in the future will be interesting to apply to analyze the effect of the deuterium depleted in cancer, and in particular in cancer cell metabolism, and we will be glad to collaborate in this field with this outstanding audience and experts that I'm glad that we have now here. And just to finalize, this is people that now is in the labs, and some of them are already in the last stage of their thesis, and some of them are just like Carlos, are just starting constructing these connections with the model of mitochondria and metabolism. Thank you very much for your attention.